CountryWide CONNECT, connecting farmers and growers to what they need to know to grow. Your daily innovative rural show with Andy Thompson and Sarah Perriam-Lampp.
Yesterday, we were talking facial eczema, and there's been a breakthrough on that today. To talk to us about this, we've got Dr. Kwasi Adusei-Fosu. He is a science team leader, Plant and Endophyte Technologies, Bioeconomy Science Institute. Dr. Kwasi, really appreciate your time on the show. Thanks so much about this facial eczema. You're talking to two South Island dairy farmers here, and we have not, between the two of us, I think we've probably seen one, maybe two facial eczema cows in our lives so far. So we're a little bit vague on this, but really appreciate your company. So you're going to have to educate us, but just talk to us about this breakthrough and what you guys have been working on.
Okay, thanks for inviting me and thanks for this opportunity. Hello to all your listeners as well. So what we've been researching over the past close 1.5 years has been how we can switch or create more of a paradigm shift from using chemicals for treating our farm animals that are suffering from the facial eczema disease. However, the fungus that causes the disease lives in the pasture, and it usually lives in the debris of the pasture. So once the animal goes into graze, picks up the spores, and it's a fungus called the Pseudopitomyces toxicarius, which was renamed by Bevan Weir and Tina Voici, all senior scientists within the Bioeconomy Science Institute. And once the animal ingests the spores, it then gets exposed to the mycotoxin, which is the sporidesmin A. And the liver, as a result, being exposed to this causes the animal to be very, very sensitive to light. That's very, very sensitive to light. And as a result of that, causing skin peeling off, reducing productivity, milk, fecundity, on and on. And as a result, having an impact on the farmers. So the main breakthrough that we've been focusing on is more of an approach that we call the RNAi technology. And I'll try and break it down for our listeners to also enjoy this conversation. So we know that the main genetic information in our body is in a bucket that we call the DNA. And for the DNA to really function, it has a functional part called the messenger. The messenger is the RNA. So the RNA then carries through the messages to say that let's produce this protein, let's produce that protein, getting information within the DNA channel. And as a result of that, we are trying to see how we can interfere with the messenger, that is the RNA carrying the information within every living cell. And so as a result of that, we are going to use what we call the RNA interference, where we are going to use more of stop button or, search and destroy, more of a mission kind of stuff within the organism. And so through the double-stranded RNA that we are going to apply, we are going to use it just as a spray-on application. It's just like buying your insecticide spray, seeing a bug or a mosquito or fly in your house, and then spraying it within your house. Because currently they've been applying fungicides in the pasture. An example of those fungicides is carbendazim, which when you come to Manawatu here, on your way to Tokoroma, there are some farm hills over there that have been applying these on the pasture during the peak season of the fungus. And as a result of that, we believe that once chemicals are being used, the premium, the value that the whole world gives to New Zealand meat will definitely go down because people are now concerned about what they consume. Besides that, we're thinking about how the social licenses of our farmers would be impacted because they are now wiping out a lot of chemicals from not being used. A good example is the zinc oxide that is being used. And as a result of that, that creates more of antimicrobial resistance. So this tool that we are talking about is a spray-on application called a double-stranded RNA that will be sprayed on the fungus in the pasture and then interfere with the RNA machinery, causing a stop button to make sure that it's not producing the mycotoxin that causes disease in the animal. And in doing so, we are presenting an alternative tool to our dairy industry. Yeah.
So, okay, a lot of information there. And correct me if I'm wrong. So did I hear you say that you've got a spray on that you can put directly onto the animal? Did I catch that?
Yes. Yeah.
So that's one. So there was one
Sorry.
vector there to interrupt it. So a spray on like an insecticide onto the animal, but then also something to go onto the pasture as well.
Yeah, we'll be applying the spray double-stranded RNA tool on the pasture, but not on the animal. Because currently the tools that have been used have been focused on treating the disease in the animal, For us, we are thinking of using the double-stranded RNA to spray the pasture to replace the alternative fungicides that are currently being used, so that we shift away from the use of fungicides in our pasture.
So when I hear RNA and stuff, I think of a little bit maybe gene editing or genetic modification. Is there any of this involved in this process?
No, thankfully, we are not going the gene modification approach. So with the RNAi technology that I talked about, explaining what a DNA is, what RNA is, what RNAi interference is, we are going to use the approach called the SIGS. It's called a 6 approach, spray-induced gene silencing. You spray the pasture, it gets to the fungus, it shuts the mycotoxin down, which is the sporidesmin, and also targeting the organism not to be able to reproduce. So more also like causing vasectomy in the fungus so that it doesn't reproduce progenies within the pasture. So we are not going the GMO approach. In case we had gone the GMO approach, we would have been using what we call the host-induced gene silencing approach. But that is far from the 6 approach that we are thinking of using, which we are working on now. Yeah.
So how far away then are we? This is research at the moment, and you're obviously very excited about the breakthrough that Dr. Bevan Weir made and you're doing here. How far away for on-farm? Are we talking 5 to 10 years, or are we closer than that?
Yes, I would say that I'm very positive on this technology because it's being used currently in other sectors. So our partners in Australia that we're working with have been able to have breakthrough for other crop protection tools. But there's a first kind of a tool that we will be using to manage fungus that causes disease in farm animals. And we are looking at around 5 years, we'll be able to get to that point from now. And we've had support from our business managers who have been able to do the market projections to help us understand the cost impact as well, and how the cost would be going down with time, because that's another aspect that we've been dealing with. And as a result of science advancing over the years, the cost for double-stranded RNA wouldn't be expensive than the normal fungicides, which are the pure chemicals that are currently being used on our farms.
So with this spray affecting the sporidesmin, is that going to have we noticed any effects on any other fungus that lives within the soil? Any positive, we have positive fungus in the soil as well that helps plants grow and that sort of thing. So are you noticing any effect on those ones with the application of this?
That's an excellent question. So as part of the project that we're currently doing, we have something I call the off-target. That's what you're referring to, where we are looking at some beneficial microbes that are within the environment where the animals graze. So as part of the pipeline that is being used currently, we are screening against, that's the genetic information that we are trying to target in the fungus against all other potential organisms that exist within that environment. A good example is the epicloe, which is in a perennial ryegrass, which is very, very important when it comes to building the resilience of our grass to biotic and abiotic stresses. So these genes have been lined side by side with the information that Bevan and Tina, Christine Voicey, were able to acquire for us over the past years. And as a result of that, we are very positive that off-target wouldn't be an issue, and I'll be able to go over that hurdle. So be rest assured that off-target is a key aspect that we are considering as part of this research we're doing. And that's a very, very good question. Thank you.
That's why I've got him there as my co-host, Dr. Kwasi. Exactly why I brought him in to ask the technical questions. Just on the product, at the moment when we do spore counts, right, we get low and high. Yeah. Would you be using different concentrations, or would you just be putting it over the entire property? Where would you know to spray and not to spray effectively?
That brings in the complementary tool that we develop in addition to the double-stranded RNA. So as part of this whole project, I would say that we have been working on a rapid detection tool that we call the loop-mediated isothermal amplification. It's just a tool that you carry to the farm and then sampling within your pasture, putting it in the suspension for it to tell you whether it's there or not. But going back to the spore count that you mentioned, the main challenge now is that most of the farmers have been following through advice based on spore counts. But most of these spores could be having low levels of mycotoxin, which is the sporidesmin A, or may not be having it. And that is another aspect of the research that we're doing. So that means that if we are telling farmers to really give the farm animals zinc oxide, we may be misleading the farmers. And that was more of a gap that we identified that we're trying to address with this rapid detection tool, where we go into the farm, we are able to detect the presence of the fungus based on the mycotoxin. Then it tells us that the population that you have here has higher levels of mycotoxin that you need to treat. Talking about the treatment, this tool then informs us that there are higher counts in certain parts of the paddock. Then we go in with a double-stranded RNA tool to do the spray-on application. So it could be a farmer understanding that the whole farm is really eaten up with or infested with Pseudopetomyces toxicarius and has to go in the direction of switching paddock and spraying it with a double-stranded RNA, or the farmer identifying the various hotspots based on the rapid detection tool that we're currently working on, which has really advanced. And we are doing this together with other veterinarians at Massey University. And so that's what I can talk about when it comes to spot spraying, why counting is not reliable to inform farmers to treat animals because some spores may contain mycotoxin, others may not contain it. So this tool then guides us to identify spores based on those having higher levels of mycotoxin and then going into treat with a double-stranded RNA.
All right, Dr. Kwasi, really appreciate your time on CountryWide CONNECT. It's pretty exciting stuff that you guys are working on. Great to be able to talk about this. And I know there are a lot of farmers who are suffering through this and a lot of stock suffer through it. It's a really serious animal welfare issue. So great work and looking forward to following this and discussing it in future.
Yes, I look forward to another invite. And it will be good to have the team here so that I get to see the wider team who are currently working on the project, because I'm not the only person. There's a bigger team behind me.
Absolutely.
And I really appreciate all the support that they're giving us.
All right. No, we really appreciate your time. All right.
CountryWide CONNECT, connecting farmers and growers to what they need to know to grow. Your daily innovative rural show with Andy Thompson and Sarah Perriam-Lampp.